Welcome back. You have just germinated. You're a seedling. You've come out of your seed. You're growing upward. You're growing upward, hoping to get to the light. But you encounter a rock. What do? Or you're a plant above ground and something has cut your top off. You're not going to be able to grow from your top, from the apical meristem at the top. What do? The answer to both these questions is hormones. Hormones allow plants to respond to their environments. These are parts of plants' control systems. So today we're going to talk about control systems. We're going to look at a few hormones. Plants have several classes of hormone. You might say 7, you might say 8, you might say 9. It's just a few. It's certainly fewer than animals have. Plant hormone action is very complicated. It has to do with where the hormones are and what the concentrations are of the different hormones. So let's have a look at some of these. So this is the study of control systems. This is the subject of control systems. You'll remember from our first lecture that I said that plants sit still and suffer the slings and arrows of outrageous fortune. That's true. As Hamlet said, plants sit still. If it gets cold, a plant does not move into the warmth. It has to do something else. It has to adjust its metabolism. It has to adjust some aspect of its metabolism for this new cold environment. So they sit still. So they've developed all of these ways to live a life of sitting still. Here's an example. Plants have lots of things that eat them. These are herbivores. Herbivores include things like caterpillars, deer, other mammals, monkeys, things that eat leaves, in particular insects. Insects are by far the biggest class of herbivores. So plants have defenses against these things. These include thorns or trichomes, which are hairs. They also have chemical defenses. These are often distasteful. They deter herbivory that way, or they might be toxic. For example, there's something called cannabinine, which is an unusual amino acid. It replaces arginine in insect proteins, and it causes the insect to die. Some plants can even recruit, so to speak, predatory animals that help defend against these specific herbivores. And here's a really neat example. This is a caterpillar that's eating a corn leaf, a maize leaf, and you can see it's wounding the plant, and it is using saliva. And this wounding and this saliva induces in this plant what's called a signal transduction pathway. This pathway causes the release of volatile attractants. So these attractants are synthesized and then released into the air, and it is noticed by these wasps that are called parasitoids. And these parasitoid wasps are special wasps that lay their eggs in other organisms. They're not quite parasites and they're not quite predators. They're somewhere in between. So they're called parasitoids. This is really common in the tropics. If you go into the tropics, you'll see lots of evidence of this. Caterpillars that have eggs coming out of them or pupae coming out of them. So this parasitoid comes along and lays its eggs inside the caterpillar, and its babies grow up inside the caterpillar. Its larvae grow up inside the caterpillar. These larvae then pupate, and you can see the pupae, the pupal cases often just sticking out the back of this caterpillar. It basically turns the caterpillar into hamburger. So this is a pretty neat way that the plant recruits something that attacks what's attacking it. So in the world of plants, there are several classes of hormone, and some of them are shown here. This is a fairly complete list, but the list continues to grow as we discover more and more about what hormones are and what different plant substances do. So we're going to talk about a few of these today, and I've listed these here with check marks and in bold. First of all, let's define a hormone. A hormone is an organic substance made in one place and transported to another, where it affects growth and other processes. In other words, a hormone is not produced in a cell and then affects that cell. A hormone, to be a hormone, needs to be made in an area of an organism and then transported to another area. How do hormones act? Well, they act, if you want to just sort of simplify things, they act in 3 steps. There is reception, there is transduction, and there is the response. So, first of all, a hormone can bind to a specific protein receptor, either embedded in the membrane or maybe it's in the cytoplasm. This receptor protein's shape, its conformation, thus is altered. This stimulates the production of relay molecules in the cytoplasm of the cell, and these relay molecules in turn trigger various responses to this original signal. And plants have a number of different signal pathways, each triggered in response to specific environmental stimuli. So this is the general overview of how hormones act, and there are only a few classes. In plants, it's the ratio of the different hormone types that really determines what a particular tissue does, how that tissue responds. So our first hormone class is abscisic acid, and this is misnamed. Abscission means falling off. Abscisic acid has nothing to do with leaves falling off or fruits falling off. Nothing to do with it. Instead, abscisic acid is involved in stomate regulation. And we mentioned this earlier, so you know about this. In dry conditions, abscisic acid, or ABA, stimulates the closure of stomates. That's because it stimulates potassium ions to leave the guard cells. And once potassium ions leave the guard cells, then water flows out of the guard cells because the water potential has been reduced outside of the guard cells. So this causes them to collapse, and they therefore close. They collapse against each other, and the stomate is closed. So abscisic acid is a very fast reaction in the world of land plants. Our next group is the group of auxins, and auxins are involved in growth in particular. They are produced in young leaves. They are produced in the shoot apical meristems. Their major functions are stem elongation, including bending toward light, or phototropism. This occurs only when the auxin is in low concentration. Auxins also stimulate the growth of lateral roots, that is root branching, and they enhance apical dominance. That is to say, they have sort of an opposite effect in the shoot compared to the root. They promote the growth of the main shoot and inhibit the growth of lateral shoots. Here's part of how auxin works. This is called phototropism, or bending toward the light, and you can see light here coming in from the left and the plant bending toward it. It bends toward it because the cells on the right side, the dark side, elongate more than the cells on the left side. This was investigated by Charles Darwin and his son Francis, and they published this in 1880, and you can see an elegant set of experiments by these two here. So on the far left, you can see a control grass seedling growing. It's bending toward the light. And the Darwins first cut off the tip of this growing grass plant. And when the tip was cut off, they found that it did not bend toward the light. Their next step was not to remove the tip, but to put an opaque cap on it, and they got the same result. It did not bend toward the light. In the fourth grass diagram, you can see the tip was covered by a transparent cap, and it bent toward the light. Finally, they put an opaque sheath down at the bottom, and it still bent toward the light. So the Darwins concluded that there was something in the tip of this grass plant that was important for the detection of light and the bending toward light. And then, a few years later, Boysen-Jensen in 1913 did a really neat experiment where he separated the tip from the rest of the plant by a gelatin block. And this gelatin block allows material to flow through it. So he found that the plant with the gelatin block in between the tip and the rest of the plant still bent toward the light. But a plant that had a mica block, something that does not allow fluid to flow between the tip and the rest, that plant did not bend. So he concluded that there was something about the flow of materials from the tip to the rest of the plant that was involved in the bending. The next set of experiments was carried out in 1926 by Fritz Wendt, and Wendt found that if he took a cube of agar and put a growing plant tip on it, and then he put that agar onto a cut plant, he could get specific results. So if you look at the bottom part of this diagram, you can see a control where the tip was cut off. There was no bending. And then an agar cube with nothing added to it. There's no bending. And then he placed an agar cube right on top. And he found that even in the dark, that agar cube that had been exposed to a growing plant earlier now caused this other plant to grow even though it was in the dark. And then he did something really neat. He put the cube only on one side, and he found that the side he put it on caused the plant to bend in the opposite direction. So if he put it on the right side, it bent toward the left. If he put this cube with this diffused chemical in it on the right, it would bend toward the left. If he put this cube with the diffused chemical in it on the left, the plant would bend toward the right, even in the dark. We now know that auxin is involved in cell elongation, and this is explained by the so-called acid growth hypothesis. And here you can see how this works. Basically, auxin increases proton activity. It increases the pumping, and therefore it increases acidity in the cell wall. And the steps here are shown on the next slide. So the first step is that auxin increases proton production, proton activity, and the acidity, therefore, is increased in the wall. Ion uptake increases in the cell wall. And then these enzymes called expansins, which are activated by low pH, start to separate the microfibrils from these other polysaccharides, these cross-linking polysaccharides. And the polysaccharides, therefore, are now more accessible to other enzymes that loosen the cell wall. This is the acid growth hypothesis, and auxin basically causes the increase in acidity of the cell wall, and therefore the cell wall can expand. And this is how the cells expand on one side of a plant more than on the other. Auxins have been used in a variety of applications by humans, and here is something that used to be more common in Canada in particular. It was called Kilex, and it's technically 2,4-D. It's an auxin-like compound. It's something that's sprayed on lawns or monocot crops, and it kills eudicots, but not monocots. It's an auxin-like compound. It is apparently harmless to humans. However, you can take 2,4-D and combine it in a 1-to-1 ratio with 2,4,5-T. Both of these are auxin-like compounds synthesized by humans. When you do this, there's a byproduct made called dioxin, and this combination of 2,4-D and 2,4,5-T is what Agent Orange is. And Agent Orange, of course, was produced by the U.S. military and used in Southeast Asia in the 1970s. The goal was to deforest vast swaths of the area, including mangrove forests and other forests. So when you mix these two auxin-like compounds together, it also contains dioxin, and dioxin is a horrible carcinogen, and it causes birth defects and a whole slew of health problems. So Agent Orange and this combination of these two auxin-like compounds is now banned, at least in the United States. Our next group of hormones are called cytokinins. Cytokinins have to do with cell division. That's what the word means. Cyto means cell, and kinins refers to movement or division. So cytokinins. They're synthesized in the roots, and then they are transported away from the roots. There are many other minor sites of production. That's true of lots of hormones. They're produced in lots of areas. The major functions of cytokinin are cell division in the roots and in the shoots. They promote lateral buds, so therefore they modify apical dominance. They promote the growth of lateral buds in the shoot, and they often act in opposition to auxins. So here's apical dominance. Here we have on the left axillary buds that are not growing, but they're just sitting there waiting, and they are inhibited by auxins. On the right, you can see that the tip has been cut off of this plant, so there is no more apex. The original apex has now been removed, and the auxins that were there are no longer coming down and flowing down through the stem. So we have now cytokinins that are promoting the growth of the lateral buds, the lateral shoots. If we apply auxin to this stump, the lateral branches would then be inhibited. So auxins and cytokinins have opposite effects here in the shoot. The cytokinins promote the growth of lateral branches, and auxins inhibit it. Here's a movie of auxins and cytokinins, and the auxins are in red and the cytokinins are in blue. So you'll see auxins being produced in the leaves and cytokinins being produced in the roots. Here we go. So the auxins cause lateral branching of roots, and the cytokinins cause lateral branching of shoots. Ethylene. The next hormone is a gas, C2H4. It is produced by most plant parts. It has several major functions. It's involved in leaf abscission, that means shedding. It's involved in something called the triple response, which we'll talk about in just a second. It's involved in fruit ripening. It's involved in root hair production. Here's a statement by the Nobel Assembly for the 1998 Nobel Prize in Physiology or Medicine. It says, signal transmission by a gas, nitric oxide, NO, that is produced by one cell, penetrates through membranes and regulates the function of another cell, represents an entirely new principle for signaling in biological systems. Wrong-o. Wrong-o, Nobel Committee, wrong by about 100 years. Ethylene as a hormone is a gas. This was discovered about 100 years before this statement. It was ignored here because this was done in animals. Ethylene was discovered in 1901 as a hormone in Russia. Around 1910 or so, emanations from oranges were found to cause premature ripening of bananas. So it was said that you should not ship your oranges and your bananas in the same containers. Now, ethylene is the most commercially produced organic compound in the world. It has lots of uses. Basically, all of the fruit you buy in a grocery store has been ripened by ethylene. So, one thing ethylene does is the stimulation of root hair production. On the left, you can see a normal seedling growing. There's a seed at the top, the roots at the bottom, and many root hairs, especially in the area right at the beginning of the root. On the right is a seedling with ethylene added, and this ethylene has caused the proliferation of root hairs. Here is abscission. Ethylene causes abscission. So on the far left is a normal leaf where auxin is produced, and the leaf is being maintained as a fresh green leaf. At some point, in this particular case, it'll be in the fall, the leaf is going to be shed. So auxin stops being produced, and therefore ethylene is produced, and the ethylene to auxin ratio therefore becomes higher. It becomes higher and higher and higher. At some point, this set of cells near the base of the petiole undergoes what's called programmed cell death, or PCD. This programmed cell death is the plant intentionally killing a set of cells. When these cells die, the leaf falls off, and you can see an abscission layer forming in this photomicrograph on the far right. Another effect of ethylene, another function of ethylene, is something called the triple response. And this is what a plant does when it's germinating and growing up through the soil and encounters something that it cannot grow past. So here's this triple response. You can see here that there are various levels of ethylene being applied to this plant, which is not facing any obstruction. It's just growing there. The triple response is, first of all, a slowing of stem elongation. Secondly, ethylene causes stem thickening. And third, it causes horizontal growth. So ethylene is produced when a young seedling is growing upward and it encounters some sort of obstruction. It encounters the obstruction, slows its growth down, thickens, and grows sideways until it gets past the obstruction and then can grow up into the sunlight. Our next effect is fruit ripening. Ethylene promotes the ripening of fruits. It's produced during fruit ripening. It is autocatalytic. it promotes its own production. It increases respiration. It allows the cells in the fruit to become ripe. That is, to become softer, to produce more sugar. Almost every fruit that you buy today in a grocery store has been stored and then ripened. It's very difficult to ripen a fruit and ship it and have it attractive to customers. Customers like fresh fruits that are not overly ripe and still firm. So fruits tend to be picked early, well before they're ripe, and then ethylene is applied. This allows the ripening to occur at set times, at specified times. So the action of ethylene in fruit ripening requires oxygen. So if you want to ripen some fruits, go ahead and put them in a bag, but it's best to put them in a paper bag, not a plastic bag, because you want a constant supply of oxygen. So if you have an avocado and it's not ripe enough for you to eat, to enjoy, or to make dip from, you can put your avocados in a paper bag, and they will ripen up fairly quickly, in just a matter of a day or two probably, depending on how unripe they are, because the ethylene that's produced will be concentrated in that paper bag. There's oxygen in there. The ethylene will stimulate the production of more ethylene, and this will ripen up your avocados, and you'll have your guacamole. See you next time.